I founded a company called Onc Therapeutics a number of years ago, and one of the key differentiating features of our technology is the ability to express potent and specific trail variants on the surface of NK cells. So NK cells can kill using perforin and granzyme, so granule-mediated killing. But another important mechanism, in fact, is by engaging the death receptors using ligands such as Fas ligand or trail that are on the surface of the NK cells. Now, there's a long history with trail in the clinic. Anybody who's interested in this knows it goes back almost 20 years. And when trail was used in a soluble form or used as agonistic antibodies against the trail receptors, the results were largely disappointing, to be honest. And this largely reflects the short half-life of these treatments and an inability to get sufficient clustering of receptors on the tumor cell. But you can achieve this by using trail on the surface of the NK cell where it's naturally found. And so by basically expressing trail in combination with a CAR, we can get a very nice synergistic response. And this can potentially mitigate against resistance from antigen escape or down modulation of tumor antigen. And also it's been shown recently in the context of CAR T cells that inefficient killing through the death receptor pathway can be associated with primary resistance to CAR T cells. So we believe that this is an important way of enhancing the killing by a CAR T or CAR NK cell. So the collaboration with Avectis our partners will involve the use of transient expression of a CD19 CAR along with this potent and specific form of trail that will target death receptor 5 in NK cells for the treatment of lymphoma and B-cell lymphoma. And because we're going to be doing this using Avectis's technology, which is called soluporation, and this allows for a very gentle, rapid transfection of cells with minimal perturbation of the immune cells. So basically the cells are ready to go straight away. And this can be done very efficiently, quickly, at low cost. It will require repeated administration of cells because of the transient nature of the mRNA transfection. But this allows us to titrate the treatment with a very important emphasis on safety. And we believe that dosing this a little bit like one would dose a monoclonal antibody gives us very good control. And in lymphoma, there isn't the same evidence that there's a necessity for persistence the way there is with ALL, for example. So we believe that with a schedule of treatment over a defined period of time with repeated doses, that we can safely and effectively administer the treatment, and that this can be done at a modest cost. I guess there's a certain amount of work that needs to be done to integrate our technologies along with this new platform for transfection, and to get the necessary IND enabling work to allow us to bring this to the clinic. So we anticipate that all of this will take in the region of about 2 years. And because we're using this methodology as opposed to relying on the use of viral vectors, we believe that we can get this to the clinic a lot faster. The lead time for procurement of GMP grade RNA, for example, is much, much shorter than would be the case for getting GMP grade vectors, retroviral or lentiviral vectors. So it's a much more rapid route to the clinic, and I think will be a very important proof of concept for Avectis's technology on the one hand, but also for our trail variant, as this will be the first time that this will be going into the clinic.